In this section, we're going to discuss glycogen metabolism. Now you can see when we look at the whole scheme of metabolism, you can see that this is the role that glycogen production and degradation plays in that. It takes 5 and 6 carbon sugars, mostly 6 carbon sugars, and converts them to polysaccharides for storage. Now, as I indicated before, glycogen is the primary storage form of glucose, and glycogen provides stored energy for short-term needs. There's two primary sites where glycogen is stored. One is in the liver. Now, the function of glycogen in the liver is to help regulate blood glucose. Glycogen can be consumed in the fasted state and produce glucose to help maintain a normal blood glucose. The other place that glycogen is stored is in the muscle. And in the muscle, it functions as a ready source of fuel for ATP synthesis during exercise. Now, glycogen is a large branch polymer of glucose. The majority of linkages are alpha-1,4 glycosidic bonds. These are ether linkages between oxygens in adjacent glucose molecules. There are also some alpha-1,6 glycosidic bonds, and these form branch points in the glycogen molecule. Now, this is important because if you had to store all glycogen molecules as long chains, they would take up a lot of room. But by creating these branch points, you're able to make glycogen into a more compact structure so that it's more easily stored, and so that the degradation is more efficient. Now, the substrate for glycogen synthesis is UDP glucose. This is a molecule of glucose bound to UDP, and it's made from glucose 6-phosphate via a glucose 1-phosphate intermediate. Now, this UDP glucose is a central molecule in a bunch of different metabolic pathways. Not only is it involved in glycogen synthesis, but it's also involved in galactose metabolism, as we've already described, in protein polysaccharide synthesis, in glycolipid synthesis, in the solubilization of drugs, and in the formation of glycoproteins, which we've described before. Now, glycogen synthase, the way that glycogen is synthesized is that there's an enzyme called glycogen synthase. And this takes glucose molecules from UDP glucose and adds them successively to the end of a growing chain. Once that chain reaches 7 molecules of glucose long, there's an enzyme called the branching enzyme. And this moves pieces of about 7 monosaccharides from the end of a glycogen molecule to the interior of the molecule to make another alpha-1,6 bond, as shown in this schematic diagram. The degradation of glycolysis, or what we refer to as glycogenolysis, is essentially the reversal of this step. There's an enzyme called glycogen phosphorylase, which releases molecules of glucose 1-phosphate from the ends of glycogen one at a time. This glucose 1-phosphate is converted to glucose 6-phosphate, which can either enter glycolysis, for example in the muscle or in brain tissue, or can be converted to glucose and released into circulation, for example in the liver, where it functions to maintain blood glucose, as we indicated before. The alpha-6-1 bonds, these branch points, are broken by another enzyme called the debranching enzyme, and this also releases free glucose. Now, as you can imagine, this process has to be carefully regulated. We only want to store glycogen in the fed state. We want to consume glycogen in the fasted state. So glycogen synthesis, which is performed by that enzyme, glycogen synthase, is upregulated in the fed state by insulin signaling and downregulated in the fasted state by glucagon, or in exercise by epinephrine. Glycogenolysis, or the degradation of glycogen, which is performed by the enzyme phosphorylase, is upregulated in the fasted state, or in exercise, and downregulated in the fed state. So this makes sense. In the fed state, we want to store glycogen, so we're going to upregulate glycogen synthesis and inhibit glycogenolysis. Whereas in the fasted state, where we want to consume glycogen, we will upregulate glycogenolysis, or glycogen degradation, and inhibit glycogen synthesis. Now, this whole thing is regulated by glucagon and by insulin. Glucagon and epinephrine activate protein kinase A via the cyclic AMP intermediate, as we described in the previous section. Protein kinase A then phosphorylates 2 key proteins. Protein kinase A phosphorylates what we call phosphorylase kinase. The phosphorylated form of phosphorylase kinase is active. And this is where it gets confusing, but phosphorylase kinase phosphorylates glycogen phosphorylase and activates it. This activates glycogen degradation. So again, the major theme is that glucagon leads to the phosphorylation of things. In this case, it phosphorylates an enzyme which phosphorylates and activates phosphorylase. And this activates glycogen degradation. And that's schematized here. So glucagon and epinephrine activate adenylate cyclase, which produces cyclic AMP, which activates protein kinase A. Protein kinase A activates phosphorylase kinase. Phosphorylase kinase activates phosphorylase, and this leads to glycogen degradation. Now, on the other hand, phosphorylated glycogen synthase is inactive, and this blocks new synthesis of glycogen. So glucagon and epinephrine lead to the phosphorylation of glycogen synthase, so it becomes inactive. And by this mechanism, glucagon and epinephrine are able to coordinately regulate glycogen metabolism such that synthesis is turned off while degradation is turned on. And that's schematized here. Again, glucagon and epinephrine activate adenylate cyclase, which produces cyclic AMP. This activates protein kinase A, which phosphorylates glycogen synthase, which is the inactive form. And this effectively blocks the synthesis of glycogen. In the fed state, insulin has the exactly reciprocal effects on glycogen metabolism. It has the effect of activating glycogen synthesis while inhibiting glycogen degradation, and it does this by dephosphorylating things. Remember that that's a common theme in the regulation of metabolism. Glucagon phosphorylates things, insulin leads to the dephosphorylation of things. So insulin works through a protein called protein phosphatase 1, and this is activated via a kinase cascade, which we won't describe. But the major thing to understand is that protein phosphatase 1 dephosphorylates and inactivates both the phosphorylase kinase and glycogen phosphorylase. The net result of this is inhibition of glycogen degradation or glycogenolysis. At the same time, protein phosphatase 1 dephosphorylates glycogen synthase. Now this activates glycogen synthase, which leads to glycogen synthesis. And that's shown again in this same schematic that we showed you before. Protein phosphatase 1 leads to the dephosphorylation of phosphorylase kinase and the dephosphorylation of phosphorylase, in effect inhibiting glycogen degradation. At the same time, it dephosphorylates glycogen synthase, increasing its activity and stimulating glycogen synthesis. Now, there are lots of defects that have been described in glycogen metabolism, and one of these we'll describe in the following clinical vignette. In this vignette, an otherwise healthy 22-year-old man presents to his doctor complaining of leg cramps. He started exercising regularly in recent days, but the exercise has been limited to very short duration because if he exercises for any length of time, he starts to have painful muscle cramps. Now, the other thing that he's noticed is after he exercises, his urine becomes dark for about a day and then returns to normal. The physical exam and laboratory testing are completely normal. So what is the disease that this patient has? This patient has a disease known as McArdle syndrome. McArdle syndrome is an example of a group of diseases that we call glycogen storage diseases. And these diseases are defects in glycogen metabolism, particularly in glycogenolysis or glycogen degradation. The clinical problems that result from glycogen storage diseases are a result of accumulation of glycogen in the tissues. This leads to cell dysfunction and cell death, and so you get loss of functional cells in tissues. The other thing that happens in glycogen storage diseases is you get metabolic defects arising from the inability of the body to mobilize glycogen stores. Now, there are at least 8 different subtypes of glycogen storage disease, and these are differentiated by the enzyme defect that's involved and the tissue specificity. But in general, we can divide these into 3 different categories. There are hepatic forms. These are the forms that primarily affect the liver. This is types 1, 3, 6, and 8. There are the muscle forms, type 5 and 7, which affect primarily muscle tissue. And then there are some miscellaneous forms, type 2 and 4. The most common form of hepatic glycogen storage disease is what we call von Gierke's disease. This is type 1 glycogen storage disease. Type 1A von Gierke's disease is caused by a deficiency of glucose 6-phosphatase. So basically what happens in this disease is that the glucose 1-phosphate, which is produced from glycogen degradation, is converted to glucose 6-phosphate, but that glucose 6-phosphate cannot be converted back to glucose. The type 1B form of von Gierke's disease is the less common form. It's caused by a deficiency of the glucose 6-phosphate microsomal transporter. Remember, I told you before in the glycolysis or in the gluconeogenesis section that glucose 6-phosphatase is found in the endoplasmic reticulum, and that glucose 6-phosphate has to be transported into the endoplasmic reticulum in order for glucose 6-phosphatase to convert it back to glucose. So in the type 1b von Gierke's disease, we're able to produce glucose 6-phosphate, and the glucose 6-phosphatase activity is normal, but we can't get the glucose 6-phosphate into the endoplasmic reticulum so it can be metabolized, and the consequence is a buildup of glucose 6-phosphate. Now, the clinical features of von Gierke's disease include things like failure to thrive. This usually presents in infants, hepatomegaly, renomegaly, or enlarged kidneys, hypoglycemia, which can lead to ketosis and or seizure. The hypoglycemia is caused by not only the defect in glycogen degradation, but also a defect in gluconeogenesis because glucose 6-phosphatase doesn't work properly. You also get hyperlipidemia leading to xanthomas, hyperuricemia leading to gout, and platelet dysfunction leading to a tendency to bleed. The way that we treat this disease is frequent feeding and a high carbohydrate diet. This decreases the dependence of individuals on glycogen metabolism. Even so, individuals still accumulate glycogen in their tissues, and mortality approaches about 50% if the patients aren't treated. Other hepatic forms are detailed in this table. These are less common, probably less important for you to understand for the USMLE. The main hepatic form to understand is von Gierke's disease. Now, the muscle forms of the disease, the most common of this is McArdle syndrome, which is what we described in the clinical vignette. This is caused by a deficiency in a muscle-specific isoform of glycogen phosphorylase. So what that means is that in the muscles, we are unable to efficiently convert glycogen to glucose 1-phosphate. And so when individuals with McArdle syndrome exercise, they're unable to mobilize their glycogen stores efficiently, and so they have difficulty producing enough energy over long periods of exercise. The type 7 form of this disease is rare, and it's caused by a defect in phosphofructokinase. The clinical features of the muscle types include painful muscle cramps with strenuous exercise. And with strenuous exercise, then you're not able to mobilize energy, and some of the muscle cells end up dying. And when they die, they release myoglobin into the circulation. And so what happens is that myoglobin gets cleared by the kidneys, and people get myoglobinuria following exercise in about 50% of the patients with McArdle syndrome. The other thing that happens is because you're unable to mobilize glucose, you get a buildup of lactate, and so they may have a lactic acidosis of a mild degree after exercise. Onset of this disease is usually later. It's about in the 20s, and the patients are otherwise healthy and have normal longevity. The most common form of in the miscellaneous category is Pompe's disease. This is type 2 disease. This is caused by a defect in a lysosomal enzyme called alpha-1,4-glucosidase, or acid maltase. In this particular disease, the cells are unable to break down glycogen in lysosomes. And this causes a massive accumulation of glycogen in many tissues, including the liver, the heart, and the skeletal muscle. And this leads to cardiorespiratory failure and death, usually before the age of 2. A rarer form is called Anderson's disease, and I highlight this one only because this is the one type of glycogen storage disease that's caused by a defect in glycogen synthesis. In Anderson's disease, there's a defect in the branching enzyme. And so what accumulates is, rather than a nice branched form of glycogen, you get long, unbranched chains of glycogen. And this is not degraded very efficiently. It accumulates in hepatocytes particularly, and this leads to progressive cirrhosis and death from liver failure, again before the age of 2. So to review glycolysis, glycogen synthesis, the net reaction for glycogen synthesis is glucose 6-phosphate plus ATP, plus the growing chain of glycogen, plus water to form glycogen with one more glucose attached to it, so N plus 1, plus ADP plus 2 molecules of inorganic phosphate. The key regulated enzyme is glycogen synthase, which is positively regulated by insulin, negatively regulated by glucagon, and associated diseases are that type 4 glycogen storage disease, or Anderson's disease, which is a defect in the branching enzyme. The net reaction for glycogen degradation is glycogen with N number of glucoses plus water is broken down to glycogen N plus 1 plus glucose. The key regulated enzyme is phosphorylase, glycogen phosphorylase, which is negatively regulated by insulin and positively regulated by glucagon. The associated diseases are the glycogen storage diseases, which we described. Von Gierke's disease, type 1, which is a defect in glucose 6-phosphatase. Type 2 glycogen storage disease, or Pompe's disease, which is a defect in acid maltase. And McArdle syndrome, type 5, which is a defect in muscle glycogen phosphorylase. This concludes the section on glycogen metabolism.